Hi, my name is Hans Bierning. I'm the Director of Product Development at Invitrogen Corporation. Luminex is a groundbreaking platform that has established itself as the standard in multiplex protein detection technology. The platform consists of a dedicated instrument and a fluorescent bead-based array. The fluorescent beads act as a solid substrate for an immunocapture assay, similar to the polystyrene plate in a traditional ELISA. The bead types differ in color and intensity, allowing each unique bead type to be part of a different immunoassay, with each protein of interest being detected by a particular antibody-labeled bead type. The amount of protein captured by this antibody-labeled bead is determined by the fluorescence intensity of the complementary detection antibody. Since you can combine these bead assays together in a single well, you can multiplex many different assays together. It's an extremely elegant solution to the need of gaining more information from small samples. Another key benefit is the use of fluorescence in the detection of the analytes. Unlike typical ELISAs, the readout for a Luminex assay is fluorescence-based. Fluorescence allows for a greater dynamic range of detection with any particular protein target, and generally greater sensitivity. The broad dynamic range of the Luminex system means exploring fewer sample dilutions to find a concentration that yields accurate quantification. Well, while it's certainly possible to optimize individual ELISA assays to achieve a high degree of sensitivity, ELISAs still only quantify one protein at a time. Invitrogen Luminex assays routinely deliver low picogram sensitivity while gathering information on over 30 analytes. There are many key interactions that can be detected when examining proteins on this scale. Also, for rare or precious biological samples like patient biopsies, there simply may not be enough material to run the multiple ELISAs required to detect all of the markers of interest. Your second thought might be, how specific are those assays? Is there any crosstalk? Can I trust the information? Absolutely. The key to specific and reliable data from the Luminex assays is the specificity of the antibody pair and the components of the buffer solution, which minimizes aggregation of the antibody mixture. High-quality antibodies are the heart of the Invitrogen Luminex system. With the acquisition of Biosource, Invitrogen is able to draw on decades of experience in optimizing antibody pairs for immunoassays. This same expertise is applied to the development of Luminex technology, since many of the same high-quality antibodies found in our ELISA assay kits are also incorporated into our Luminex assays. When we develop antibody pairs, we make great efforts to eliminate cross-reactivity and to validate their performance in a range of real-world samples. What this all means is that our customers can easily switch from ELISA to Luminex without having to worry about re-optimizing their system. They can expect the same data from both platforms. In fact, we provide a conversion factor for easy data comparison between platforms. Additionally, all of the same great performance claims made about commercial ELISAs also apply to Luminex assays, giving the user confidence in the assay. Lastly, the standards we use in our assays give you that extra bit of confidence to make the switch to Luminex. We take great care to mass calibrate the standards to ELISAs and to make them physiologically relevant. They're tested for linearity with natural samples and for cross-reactivity with the bead assays it should not react with. All in all, you can really count on these assays to accurately detect proteins in complex mixtures. Since its introduction, Luminex technology has been used throughout basic research fields, particularly in areas related to signal transduction and to cytokine protein expression. It's also used in biotechnology and pharmaceutical research, where screening biomarker panels in patient or test animal responses to treatment is a key part of evaluating drug performance in preclinical trials. Well, just think of it, being able to simultaneously examine 5, 10, or 20 biomarkers in one 50 microliter cell or tissue sample. The complexity of the interactions and the ease with which this can be revealed is astounding. It would take days of effort, much greater expense, and loads more sample to achieve the same results using individual ELISA assays. That one person could do the same thing from a single sample in tens of minutes. That's the real power that Luminex represents. You can expect Invitrogen to continue to release novel assays to expand the analytes available and to push the multiplexing envelope to bring you continued productivity gains and increased experimental insights. Well, it was nice chatting with you. Call us if you have any questions about this exciting technology.